you so much. Hello, everybody. My name is Sasha. I'm a PhD student in the Schlein Lab at the Hospital for Sick Children in Toronto, Canada. Okay, so of course I work at Sick Kids, and although we've made tremendous strides in the past couple decades treating pediatric cancer patients, I say we pay just as much attention to life past the initial disease. Indeed, childhood cancer survivors face really significant lifelong risks and comorbidities, and particularly in the first 5 to 20 years post-initial diagnosis, almost 80% of extra deaths are attributed to recurrence or relapse of the initial disease. So luckily, we've already heard a little bit about mutational signatures, so I won't define these for you, but I would like to bring your attention to those signatures attributed to different types of chemotherapy. This is really what I'm interested in, so we can see the effects of different types of mutagenic chemotherapy, including platinum agents like cisplatin here, and the resulting mutational spectrum upon sequencing. And for me, this mutational signature is like a battle scar. It tells me that these cells have not only been exposed and affected by the chemotherapy, but they've gone on to proliferate and expand up to our sequencing limit. And this battle scar means it's a type of resistance felt in the genome. And indeed, I truly believe that these mutational signatures are much more than just mathematical constructs. Already, we've begun to bring them into the clinic for other uses. For example, as a readout of HRD for ovarian and breast cancers. But what about pediatric cancers? I want to emphasize that both in our groups and others, we've found these therapy-associated mutational signatures in specifically relapsed pediatric genomes, both by our group here on the left and others on the right, and I want to know more. I want to know when and where we feel the effects of chemotherapy in relapsed pediatric genomes. Ultimately, at the end of the day, how we get from a primary untreated tumor to a horrible aggressive relapse showing one of these therapy-associated signatures. And to do so, I'm going to marry the mutational signature analysis with our clonal evolution reconstruction. So in order to do this, I devised a pipeline, which works from paired whole genome tumor normal. And using our in-house pipelines, I first call mutations, etc. And then using some tools with the experts in the audience here to reconstruct the evolutionary histories. Now, normally we do mutational signature analysis at the bulk level. So we take all of the tumor mutations and run the signature on that, but I do something a little bit differently. Once I've made my trees, I go into every single tree node, every single little subpopulation, and from there I do the mutational signature analysis. This is in order to learn how the mutational processes are changing over the tumor evolution. And you can imagine how this gets interesting when we have multiple samples and so begin to see changes across space and time. So in order to investigate these rare pediatric tumors, I had to get my grubby little hands on as much data as I possibly could. And so I combined 10 different pediatric cohorts from around the world, and I immediately discluded any patient samples that were just a single primary untreated tumor. This is of no interest to me. This left me with about 1,700 patients broken down by the primary tumor diagnosis on the right-hand side here, and also includes almost 600 multi-sample cases, and of course, my extremely important clinical treatment metadata wherever possible. So let's look at some results. I'm going to start with my therapy mutational signatures. So here I'm showing you the profiles from a number of different signatures we have seen associated with the chemotherapies, and I've broken it down into alkylating agents on the left and antimetabolites on the right. In a subset of about 230 patients with really detailed clinical data, I saw that about 40% of platinum-treated patients had the associated signature, and this contributed about 30% towards the overall mutation burden. In contrast to antimetabolite signatures, which I saw much less frequently and contributing fewer mutations. A third piece I'm going to add into this is a slightly different type of signature. Now we're not just looking at the SNVs, but this is an indel-based signature for radiation. And in this, a whopping 85% of my irradiated samples show the signature. What I think is going on here is that the presentation of the signature in the genome is reminiscent of the original treatment's mechanism of action. So the alkylating agents, which can directly damage, like I showed you the platinum, versus the antimetabolites, which rely on active cell cycling. And we know in the clinic to be a slower chemotherapy to act. It takes time to incorporate these nucleotides versus the massive, inescapable damage of radiation, and thus the resulting prevalence and also contribution in the genome. Next, we're going to marry the signatures and the clonal evolution. Let's take a look at the example output from my pipeline. So here I have for you both a primary and a relapsed tumor sample. This patient was originally diagnosed with malignant rhabdoid of the kidney. 8 months later, after a number of different chemotherapy cocktails, unfortunately, we see a relapse. And so I have the 2 sequenced tumor samples. I've reconstructed the tree, so we read the trees from left to right through evolutionary time, and you can see that there are subpopulations that are either shared, both colors, or are unique to one of the 2 samples. Finally, we overlay my mutational signatures, and now it gets interesting. I can pinpoint SBS35, my platinum signature, very specifically to nodes on the tree, and here they appear to be one after the other in this nice little lineage. But what I'd really like you to take away from this is that this lineage, 9, 10, and 11, actually had ancestors that were present at the time of diagnosis. So for whatever reason, there's something special, some survival fitness advantage about these subpopulations that has allowed them to go on and proliferate. My pipeline also gives us a lot of other information, of course, cancer cell fraction, degree of clonality, as well as the mutational size of each of these subpopulations. And again, my 9, 10, and 11, my green stars here, you'll notice are the most mutationally large subpopulations. So the therapy, which is super mutagenic, is actually contributing and making these bloated subpopulations in parts of my tree. Now, analyzing hundreds of tumor samples, I begin to see some patterns emerge. So let's look at osteosarcomas here, which we know in pediatric cancer often end up metastasizing to the lungs, pulmonary mets. And I'm fortunate here to have a gorgeous multi-sample case in which I have sequencing from 3 different mets that are completely different spatial sites around the body. And we see that we end up developing SBS35 platinum signature in every single one of these mets. So this systemic chemotherapy is traveling around your blood, and all separate mets and all separate sites have ended up converging upon this same SBS35 resistant phenotype. Zooming out a little bit of the osteos, I just showed you this case on the left, but I want to show you some more because it's the same case. All these post-treatment mets end up developing the signature, but you'll notice that they do so at the very ends of the tree. It's usually the last 1 or 2 subpopulations here, and you can tell that a lot of evolution for the metastasis has already happened. So I wonder here if whatever process is driving the metastasis, they've already seeded, planted, bifurcated, expanded, and then later on do I feel the effects of the chemotherapy. So I've thrown a lot at you. Unfortunately, I'm out of time, but I hope that I've persuaded you of the utility of this method to be able to see really incredibly the resolution and exactly where we feel the effects of some of these chemotherapies, that in fact, perhaps one day we could look to predict which parts of the tumor are more likely to go on to become resistant and relapse, that it's sometimes changing the topology of the tree, and that we begin to see patterns across the drug types or the tumor types. And at least for osteosarcomas, we're learning that these are perhaps different mechanisms going on. Ultimately, I want to contribute to, we're trying to get these patients towards precision medicine and to ultimately predict resistance before it happens. I want to improve the quality of life for these pediatric cancer survivors. Thank you.
Excellent talk.
Thank you so much.
Excellent talk.
Hi, Sasha.
Hello.
Hi. Thanks for that talk. That was really great.
Thank you.
We love all that stuff, obviously. You brought up the question about why is it the leaves of the trees. And I'm wondering if you know about the clonal fractions of the interior nodes. You might just be detecting those because you can,
Right.
but like the large part of the primary is actually the leaves.
What do you mean? Is it like a truncal versus a...
No, what I mean, yeah, they're not truncal. You just the composition of the primary is primarily made up of the clones that are at the leaves of the trees, and the internal nodes are a history of the evolution, but they no longer exist in the primary.
That's right.
Is that what you're seeing? Like, or?
It depends on the case. Sometimes, I do see a lot of the primary tumor remains. But some cases we do have such an enormous sweep that I have such a large gap. I have the very early ancestors, then I have what I have now. And in between, not much of it is actually physically represented in the tumor. It's just what we've tracked back, but it isn't physically there. Let's talk later.
Yeah, if